Today, explore Biology Encyclopedia. 30 Most Important Biology Equations and Formulas. Part 4. Introduction. Welcome to LSKSM Vlogs. Today, explore 30 most important biology equations and formulas, Part 4, and discover essential equations used to understand biological processes, genetics, ecology, chemistry, and cellular science. Let's continue our educational journey and learn how these important formulas help explain the fascinating mechanisms and patterns of life. One. ATP hydrolysis. ATP hydrolysis releases energy by breaking a high-energy phosphate bond. This energy powers cellular processes such as active transport, movement, biosynthesis, and signal transduction. Two. Hardy-Weinberg equilibrium. The Hardy-Weinberg principle describes a theoretical population in which allele and genotype frequencies remain constant. It provides a foundation for studying genetic variation and evolutionary change. 3. Allele frequency. Allele frequency represents how often a particular allele occurs within a population. Measuring allele frequencies helps scientists understand genetic diversity, inheritance, and population evolution. 4. Exponential population growth. Exponential growth occurs when a population increases rapidly under ideal conditions with abundant resources. The model demonstrates how populations can grow continuously when environmental limitations are minimal. Five. Logistic population growth. Logistic growth describes population increase when resources and environmental conditions impose limits. Growth slows as the population approaches the environment's carrying capacity. 6. Shannon Diversity Index. The Shannon Diversity Index measures biodiversity by considering both species richness and relative abundance. It is widely used to compare diversity across ecosystems and biological communities. 7. Photosynthesis. Photosynthesis converts light energy into chemical energy, allowing plants, algae, and some microorganisms to produce organic compounds. It also releases oxygen and forms the foundation of many food chains. 8. Cellular respiration. Cellular respiration converts nutrients such as glucose into usable cellular energy, primarily in the form of ATP. It involves pathways including glycolysis, the citric acid cycle, and oxidative phosphorylation. 9. Relative growth rate, RGR. Relative growth rate measures the increase in plant biomass relative to its existing size over time. It is useful for comparing growth performance among plants under different conditions. 10. Net assimilation rate, NAR. Net assimilation rate represents the increase in plant biomass relative to its photosynthetic leaf area. It helps evaluate how efficiently leaves convert light and carbon dioxide into new plant material. 11. Water use efficiency, WUE. Water use efficiency describes how effectively plants use water to produce biomass or assimilate carbon. It is an important concept in plant physiology, agriculture, and drought research. 12. Germination percentage. Germination percentage indicates the proportion of viable seeds that successfully germinate under specified conditions. It is commonly used to evaluate seed quality, viability, and reproductive performance. 13. Relative water content, RWC. Relative water content measures the hydration status of plant tissues compared with their fully hydrated condition. It is an important indicator of plant water balance and drought stress. 14. Mitotic index. The mitotic index represents the proportion of cells undergoing mitosis within a population of cells. It is commonly used to study cell proliferation, tissue growth, and the effects of environmental or chemical factors. (15) Cell doubling time. Cell doubling time is the period required for a population of cells to double in number. It provides valuable information about cellular proliferation and growth rates in laboratory cultures. (16) Cell growth rate. Cell growth rate describes how quickly a cell population increases over a given period. It is widely used in microbiology, cell culture, biotechnology, and cancer research. (17) Surface area to volume ratio. The surface area to volume ratio influences how efficiently cells exchange materials with their surroundings. Smaller cells generally have a higher ratio, supporting faster transport of nutrients, gases, and wastes. (18) Mutation rate. Mutation rate measures the frequency at which new genetic changes arise in a population or during replication. Mutations provide genetic variation and can influence evolution, disease, and adaptation. (19) Genetic distance, Nee's formula. Nee's genetic distance estimates the degree of genetic difference between populations or species. It is widely used in population genetics to study evolutionary relationships and genetic divergence. (20) Inbreeding coefficient, F. The inbreeding coefficient measures the probability that two alleles in an individual are identical by descent. It helps researchers assess genetic relatedness, inbreeding, and potential loss of genetic diversity. (21) Selection differential, S. The selection differential measures the difference between the average trait value of selected individuals and that of the original population. It is an important concept in quantitative genetics and artificial selection. (22) Response to selection, R. Response to selection describes the change in the average trait value of a population after selective breeding. It is influenced by factors such as heritability and the strength of selection. (23) Michaelis-Menten equation. The Michaelis-Menten equation describes the relationship between enzyme concentration, substrate concentration, and reaction velocity. It is fundamental for understanding enzyme kinetics and catalytic activity. (24) Henderson-Hasselbalch equation. The Henderson-Hasselbalch equation relates the pH of a solution to the concentrations of a weak acid and its conjugate base. It is especially important for understanding biological buffer systems. (25) Nernst equation. The Nernst equation calculates the equilibrium potential of an ion across a biological membrane. It is fundamental to understanding nerve impulses, membrane potentials, and cellular electrophysiology. (26) Osmotic pressure, Pi. Osmotic pressure is the pressure required to prevent the movement of solvent across a semipermeable membrane. It plays an important role in water movement, cell volume regulation, and plant physiology. (27) Bacterial generation time. Bacterial generation time is the time required for a bacterial population to double under specific conditions. It helps scientists analyze microbial growth, reproduction, and culture dynamics. (28) Colony forming units, CFU. Colony forming units estimate the number of viable microorganisms capable of producing colonies on a growth medium. CFU measurements are widely used in microbiology and microbial population analysis. (29) Antibody titer. Antibody titer indicates the concentration or relative level of specific antibodies present in a biological sample. It is commonly used in immunology, diagnostics, vaccination studies, and infectious disease research. ELISA concentration calculation. ELISA concentration calculations use enzyme-linked immunosorbent assays to determine the amount of a specific antigen or antibody in a sample. Standard curves and absorbance measurements help estimate unknown concentrations. Conclusion. Thank you for joining LSKSM Vlogs for Biology Encyclopedia 30 Most Important Biology Equations and Formulas, Part 4, and exploring essential mathematical and scientific concepts in biology. We hope these equations and formulas help you better understand biological processes and their real-world applications. Stay connected with LSKSM Vlogs for more exciting biology encyclopedia topics and educational discoveries in Part 5. These videos are not only for the current generation. They are a humble effort to preserve knowledge, history, science, and human achievements for future generations. Thank you to everyone who supports and encourages this journey of building a digital library of learning for tomorrow. If you enjoyed this video, please subscribe to the LSKSM Vlogs channel, like, comment, and share it to help pass on knowledge to future generations. Your support and encouragement mean a lot to us. G. Sobhanadri Rao, LSKSM Vlogs.